Hey guys, so welcome. This is Sir Shafar Khan on your screen. I hope you're all well. Let's get started with Inheritance 10.2 IB Biology. Okay, we're going to go briefly over dihybrid cross first, and then we're going to study about linked and unlinked genes. Okay, and yeah, they're slightly about their relationship with Mendel's laws of inheritance. Okay, so let's begin. So let's start by doing dihybrid cross. Okay, so what is a dihybrid cross? I'm assuming you've already completed monohybrid cross. Okay, and now we're going to start with dihybrid cross. So di actually means two. Okay, so it has something two in it. Okay, so it has to do with two things. Okay, what are those two things that we are dealing with? So it's actually a type of cross which is related to the inheritance of two sets of genes. Okay, two genes basically are inherited together in a dihybrid cross. Okay, now these genes. These may or may not be linked, but over here in this example, we are assuming these are unlinked genes. By unlinked, I mean that they are present on different chromosomes, okay, different chromosomes, not on the same set of homologous chromosomes or not on the same chromosomes, okay? They are located on different chromosomes. That is what unlinked genes mean. So linked would mean that they are located on the same chromosome, right? But over here, we are not talking about the linked ones. We're talking about the unlinked ones first, okay? So dihybrid cross is the type of cross in which we are investigating two genes which are inherited together. Okay, both of them, the inheritance of two genes. Well, not necessarily inherited together, but yeah, their inheritance is investigated over here in this type of process. It's called a dihybrid cross. Okay, so we have a parent generation right over here in which we have, okay, capital R and capital Y. Okay, these R and Y are representing two traits of seeds. Okay, over here capital R or R refers to round or wrinkled genes, whereas Y refers to the color. Okay, so this is the shape of the shape of the seed, and this is the color of the seed. Okay, this is the seed color, and that's the seed shape. Okay, and color, capital Y represents yellow color, small y would represent the green color, and capital R represents the round shape, whereas the small r a small r will represent a wrinkled shape. Okay. So now we have capital R, capital R, capital Y, capital Y. What does this term over here mean? Capital R is representing the homologous pair of chromosomes, right? The two alleles of the same gene in the homologous pairs. And capital Y is representing a different pair with a different set of genes in it, right? And this is entirely, we're just assuming, considering only two properties or two traits of the chromosome of the parent generation over here. Okay. Now, and same applies over here as well. Now, since they are uppercase and lowercase, uppercase would mean that they are actually dominant. Okay, the round shape is dominant, the yellow color is dominant, whereas the lowercase would mean that the small r would be recessive and small y would be dominant. Small y would refer, meaning green color is recessive and wrinkled shape is also recessive. Okay, so when you get the F1 generation by crossing these two parents together, okay, the F1 generation or the first filial generation that you get would have this genotype, right? This is the genotype that you will get after crossing these two, okay, the F1 generation. And the reason why you'll get this is already mentioned in the previous studies as well, right? Because capital R and capital Y are the only combination that you will get from this parent in one gamete, okay? And small r and small y is the only combination that you will get from from all the gametes that are from this parent, okay? Basically, homozygous recessive and homozygous dominant would give you only homozygous dominant and homozygous recessive genes. Okay, now these in the gametes, these gametes will cross together and they'll form heterozygous dominant F1 generation. Now this F1 generation is self-pollinated, that is, it crosses with itself. Now this F1 generation or the capital R small r, capital Y, small y generation, which is your F1 generation, will give you four different types of um, gametes, right? Because now see if you consider these two. Okay, so according to Mendel's law of independent assortment, out of these two R's, you can only get one, right, in the gamete. So you get just one capital R, and out of these two Y's, you'll only get one. So a capital R and capital Y is one combination that you can get. Then a capital R with a small y is a second combination that you can get, right? Then a small r and a capital Y is a third combination, and then a small r and a small y is another combination. So these are the four possible combinations that you can get for two different sets of genes over here. Okay. And these four combinations will be from both the sides, from both the parents, right? Because both the parents had this genotype. Okay. Now in this Punnett square that you can see over here, capital R and capital Y, capital R and small r and small y, and small r and capital Y. All of them are in equal proportions, right? 1 is to 1 is to 1 is to 1, right? So this is the genotypic ratio that you will have, 1 is to 1. Okay, so they are all equally likely um, to occur, all right? So 1/4 is the probability of getting all of these together. Similar would be the case from this side of the parent as well, right? Capital R, small y, capital Y, and all of these, right? When this gamete and this gamete fuse together, they'll give you this offspring, right? Capital R and capital Y, homozygous, right? Meaning it is round and yellow in color. Similarly, round and yellow in color again with this genotypic ratio. Okay, just write it down, and all of these have been written till here. Okay, you can see that there are wrinkled ones here as well. Okay, and that's because you get homozygous R genes, right? The lowercase r genes over here, and they are recessive. Whereas the yellow color is still there, so it's still yellow in color. So you get all of these combinations of offsprings, right? Now, if you look at the phenotypic ratio, the phenotype that is the physical appearance of all of these, the phenotypic ratio, what would the phenotypic ratio look like? So in the phenotypic ratio, you can get either yellow and round shapes, right? As seen over here, 1, 2, 3, 4, 5, 6, 7, 8, and 9. 9 of these are yellow and round. Okay, just the physical appearance. If you look at the physical appearance, so 9 of them are yellow and round. 3 of them are green and round, right? 3 of them are green and round. If they're green and round, they're right over here. Then 3 of them are wrinkled and yellow. They're wrinkled and yellow. You can see them right over here. And then one of them is green and yellow, right here. One of them is green and yellow. This is the typical or the famous phenotypic ratio that you will get. This is the Mendel's ratio, 9 is to 3 is to 3 is to 1. Okay. And this is the typical phenotypic ratio that you get when you self-cross a heterozygous dominant um, individual. Okay. Oh, sorry about that. Okay. So 9 is to 3 is to 3 is to 1. That's the phenotypic ratio that you get. Genotypic ratio, of course, is going to be different. Okay. Now, if you let's read through this text that I've compiled as well. So the inheritance of two genes is investigated together in dihybrid crosses because the two genes for seed color and seed shape are not on the same chromosome. That is, they are not linked together. The chance of inheriting S or S does not affect the chance of inheriting Y or Y. Okay, it's written S over here. What it means is it's referring to this capital R and small r actually. Okay, that's a mistake. Meaning round or wrinkled. Okay, capital R is round and small r is wrinkled. So inheritance of these would have no effect on the inheritance of Y. Therefore, they would assort independently and they would follow Mendel's law of independent assortment. Okay, this theory, the two alleles of the two genes pass into gametes without influencing each other. Okay, they're not linked. It's called independent assortment, and independent assortment is possible only in unlinked genes if they are not on the same chromosomes. If they are unlinked, okay, if they were on the same chromosome, then they would not assort independently unless and until crossing over occurs, of course. These are unlinked genes. If independent assortment is occurring, then we should expect a typical phenotypic ratio. Okay, and this is a typical phenotypic ratio. And if you get this phenotypic ratio, then, for example, if you have an unknown case, an unknown individual being crossed with another individual, and you don't know, and you're trying to investigate if whether the genes are linked or not, you can actually do a self cross for those genes, right? So this typical phenotypic ratio is obtained when independent assortment occurs and the genes are not linked together. Okay, so if the phenotypic on the exams, you might get a different question in which the phenotypic ratio is not the same. It's not 9:3 to 3:1, and that is the examiners usually actually they love to ask. Why is that the case? Why don't you get this typical ratio in that example? So if you don't get the typical ratio, then the possible explanation for that is the genes might be linked together. Therefore, they are not following independent assortment. Okay. Okay. Yeah. So we have this classical example also mentioned here, and it's related. Let's move on now. The next part. Let's look at some sample data that is following independent assortment. Okay. And this is the data of Mendel. Okay. The predicted number is given right over here. Okay. And these were the actual numbers obtained. So you can see that the actual numbers were quite close to the predicted numbers. Actual numbers of these phenotypes. Okay. So nine yellow round. So yellow round. What man in this experiment, what we do is we take a whole like a big number, a big sample of um round and yellow seeds. Okay. Which are heterozygous, and you cross them with one another, right? You cross them and you get this number 315, 108, 101, and 32. And these numbers are actually very close to the predicted numbers. Okay, they are in the same ratio of 9:3 to 3:1. So if they are very close, then that means Mendel was right. Okay, Mendel's law of independent assortment was right. That means that the shape, the gene that determines the shape, and the gene that determines the color, they assort independently and they are not influencing one another. They're not linked together. Okay, now important. We assume that the genes are unlinked. Okay, on different chromosomes. This is what unlinked means, but and they're inherited independently. um, Mendel's law of independent assortment is also known as Mendel's second law. This is in accordance to Mendel's second law that the genes are unlinked and they'll assort independently and they'll follow this 9:3 to 3:1 ratio. But if you don't get this ratio, then that means that they are not assorting independently and the genes must be linked. Okay. But the actual results are different. So in that case, I've answered your question. Maybe the genes are can be on the same chromosome in that case. So there are exceptions to Mendel's law. Okay, and let's look at them right over here. What does independent assortment look like? Okay, so we have a few examples over here of a few genes. Okay, now let's assume, let's look at the left side over here. We have capital A, capital A. Means this is a homologous chromosome. Okay, and you have this gene A, and it has its two alleles right over here. And then you have this gene small a, and then with its own alleles over here. So now during gamete formation, what would happen? One of these two would go into the gamete, right? One of these two A's, and again, both of them are the same, so they'll give just A, right? Nothing else. Similarly, from here as well, the gametes would get the small a. Let's look at a different chromosome now. Okay. This is a different chromosome. This is a different chromosome with its own homologous pair. So now over here, look at the second pair of homologous chromosomes. Now this is a different gene. Okay, it's not on the same chromosome. This is a different set of chromosomes. Okay, with a different gene. And from here as well, during um, gamete formation. So this left side will give you one gamete. Okay, the right side would give you the other gamete. So because in gametes during anaphase, they'll divide into halves, right? So out of these two sets, out of these two alleles, basically you'll only get one gene. So, and that is A. You can only get A in the gamete. And out of these two Bs, you'll only get one B. So AB is one genotype of the first gamete. Then the second gamete that is produced from here is gonna only have one A or one small a and one small b, right? There you go. You have it. They could line up like this during the metaphase, right? Or they could line up like this as well, right? The right side combination. Meaning the capital A over here will have a small b with it, right? Capital A and a small b. Both of them would go into the same gamete, right? The same applies over here, right? Both of them will give you the same type of gamete. And then the last combination, small a can go with the capital B. It will give you this type of gamete, and same applies here as well, right? Now, these are the four different gametes with their um, genotypes that you will get. This is what independent assortment looks like. Okay, meaning inheritance of A does not affect the inheritance of B. Meaning with capital A, you can have a capital B or a small b. It doesn't matter. See, you have a capital A and with a capital B or capital A with a small b. You can have both. This is independent assortment. Whereas with a small a as well, you can have a small b or a capital B. It doesn't matter. Okay, this is totally uh, by chance. This is based on probability. Okay, and it's independent assortment. That's what independent assortment looks like. Because the orientation is random, right? You can have all sorts. But what does gene linkage looks like? And by the way, over here in independent assortment, um, over here you'll end up getting the typical Mendelian ratios. Okay, now let's move on. Meaning 9:3 to 3:1, the typical phenotypic ratio. Now let's look at gene linkage. Now gene linkage actually means that the two or more genes are located on the same chromosome. Okay, not on different chromosomes, rather on the exactly same chromosomes. So now if you have gene A on one homologous chromosome, you'll have the gene B on the same chromosome. So then both of them would be inherited together, right? So the probability of A inheriting with B would differ, right? Meaning whenever A is inherited into the gametes, B would be inherited along with it, okay? Whenever A is inherited, B would be inherited alongside. Similarly, when small a is inherited, the small b would be inherited with it. Okay, you cannot have a capital A small a and a capital B in this case until and unless something else happens, right? That is, crossing over takes place. You will have a small a and a small b, and that's it. You cannot have more combinations in this case. These alleles do not assort independently. Okay, or if you have a small a and a small b over here, or a capital A and a capital B over here, this is where crossing over can take place, right? Or if a second combination, meaning this combination is only possible in a very special circumstance. But before, let's assume they assort independently. So in an independent assortment, these are the only gametes that you will get. You cannot have more gametes in gene linkage, right? A capital A will only have capital B and that's it, nothing else, not a small b. A small a will only get small b. But if you perform the experiment, you might find some different results as well. And that is a very common exam question. That why do you get a different result even after gene linkage? That occurs usually when crossing over has taken place, meaning this capital B and this small b, they switch positions. and that the crossing over does occur in meiosis, right? During variation to cause variation. When capital B and small b they switch positions and the sister chromatids become non-sister chromatids, then so capital B comes here, small b goes there. So now in this case, you can have a small a and a capital B, which is not a combination given by the parent generation, and then you can have a capital A with a small b, right? Now, these are two new combinations that you got, which are different from the parents' combination. These new combinations over here, they have a very special name. They are called recombinants, okay? Because they have been formed after combination, after recombination, after crossing over, okay? Unless and until crossing over takes place. Now, what is crossing over? Let's study about crossing over. Crossing over is the exchange of segments between the chromosomes that usually occurs during meiosis. Okay, but again, crossing over is very random and it occurs by chance, and not many chromosomes undergo crossing over. So it's roughly like 6 to 8% of the chromosomes. They can give you recombinant results. Okay, but not more than that. Majority will still have the parent genotype. Let's look at the crossing over here as well. Some recombinants can form this way. Okay, for example, if you had this small b over here and a capital B over here, the capital B and small b can switch over. Okay, a small b from here goes there, and the capital B from there comes here. So capital B is here, small b is here. Okay, and chiasmata formation occurs during meiosis, right? That's where crossing over occurs. Chiasmata is this attachment point. The overlap point is called chiasmata, right? So the points where the homologous chromosomes they cross over, and crossing over produces recombinants. Okay, but the ratios won't be the ratios predicted by Mendel, okay, because they are less common. They can only occur as a result of chance. Okay, so this way we finish off gene linkage. Okay, so linked genes they'll always be inherited together, whereas the ones which are unlinked they won't be inherited together. They'll be inherited under according to independent assortment, Mendel's law of independent assortment. Okay, but the gene linked genes they'll be inherited together, and they will not follow Mendel's predicted ratios of 9:3:1. Okay, so in this module we finished off the hybrid cross briefly, just a review of the hybrid cross, then the ratios. Okay, and then we talked about gene linkage and independent assortment. Independent assortment means that the two genes are on the different chromosomes, and inheritance of one gene does not affect the inheritance of the other. So one gene can have multiple, like four combinations. Okay, but over here in gene linkage, one gene is linked with the other, so it can only have combination with that linked gene, and that's it, nothing else. Unless and until crossing over takes place, which is very, very less common compared to the independent assortment. Okay, and crossing over will only give a few genotypes of that combination of recom, and these crossed over combinations are called recombinant. Okay, that's it. Thank you so much for being with me.